get started for today? Okay, I think we pretty much about reached steady state, so let's begin. All right, so welcome everyone. For those of you who are here the first time, good to see you again. For those of you who are not here, welcome to the BIOME Summer Webinar Series. My name is Alan, and Colin is not here today, but he will be giving the 4th lecture, so he'll definitely be there then. Today is the 2nd, and today we'll be talking about some DNA landmark studies, so just more DNA stuff. Also, almost July 4th, so wishing everyone happy Independence Day this weekend. Okay, any questions before we begin today's talk, today's webinar? All right, as a recap from last time, we were talking about DNA as the stuff of heredity, right? It explains why we look like our parents. It explains, why we have traits, but then we kind of ended off there. We were like, well, we have this stuff called DNA, there's a sequence inside of us. It's like a code. If any of you, use Python or R or C++, it's like a code, right? Inside each of our cells. But again, I feel like we're still missing that link, right? Where is the link between the sequence and biological traits? And we ended up here. So we ended up we have a whole bunch of A's, C's, T's, and G's. How does this actually encode traits? And so again, just like last time, go to this link, polev.com/AlanTong472, and you should be able to enter in your responses. Okay, there we go. So I see some coming up. I think this was from last time, actually. So DNA to RNA to protein. Yes. Transcription and translation. Yep, exactly. That's exactly what this is describing, right? Yes. More things about proteins. So it seems like people already have some background about how DNA encodes traits, which is good. It's good. And my hope for these webinars is that people of all levels can learn something. You might know already about transcription and translation. You might not know anything about DNA or RNA or proteins. And I hope that everyone takes something away from today's session. It also controls the rate of expression of genes. Yeah, so now we're going one layer deeper, right? So people are saying, well, genes can be expressed. Well, what's controlling the expression of those genes? Well, more genes. It's a chicken and egg situation, right? It gets kind of mind-boggling that way. Yep, more transcription and translation. Central dogma, exactly. And so I'm seeing a theme here, right? So central dogma, whoever put this down, what they're talking about is exactly that process in the very beginning we saw, DNA to RNA to protein, the processes of transcription and translation. And if these terms don't mean anything to you, that's completely okay. Because we'll go over them. So don't worry. Okay, I think we've got a good response. And it seems like people are pretty on board, right? So people are saying that the processes of transcription and translation, this central biology dogma, is what allows DNA to code for traits. Now, typically I like to separate the word from, the word dogma from science, right? If something's dogmatic, it's like, oh, it's so central, you can't break it. I don't like that word. I think the term central dogma is a misnomer. Because the central dogma is DNA goes to RNA goes to protein, and we'll talk in depth about that. But can anyone think of exceptions to that dogma in quotes? And feel free to type in the chat. So Rohan said ribozymes. What do you mean, Rohan? Do you mind elaborating a little bit? So ribozymes are enzymes that are made out of RNA, and they're functional RNA. Yes, yes. And so the idea is that not all RNA gets turned into proteins. Is that what you're getting at, the idea? Yeah, exactly. So sometimes it stops at RNA, right? RNA has a whole host of functions. And Eileen says reverse transcription. So we have transcription. There's also reverse transcription, right? When you go from RNA back to DNA. HIV does that. And Ramya says when there is a mutation in the DNA sequence, it codes for a stop codon so that the protein is not fully produced. Right. DNA can have mutations, and that can have downstream effects. It can affect the RNA and the proteins. And so I guess it would still follow the flow of the central dogma. But yes, some mutations can also occur that might cause a break in the chain, perhaps. Very good. So we call it central dogma, but know that it's not really a dogma, right? And whoever put this one, just wait. I think you know what I'm going to be talking about, but just you wait about environmental factors. Because right now, things seem to be set in stone, right? We have a genome. We have A's, T's, C's, and G's in a certain sequence inside all our cells. It seems like, oh, life is deterministic. Of course it's not deterministic, right? There's all kinds of random, stochastic, chaotic things going on in our world, right? It's not like your DNA has a sequence that told you to dye your hair pink the next morning, right? It doesn't happen. There's environmental factors that cause you to have different traits, right? Your pink hair color that you dyed isn't in your DNA. That was an environmental factor. So very good. Okay, so let's move on. Oh, whoa. Did it turn big for all you just now? Let's see. How do I move on? Come on, next. Okay, here we go. So here's what everyone's been saying in diagram form. You've got your DNA. DNA, as we mentioned, deoxyribonucleic acid. It's in the nucleus of the cell, right? DNA gets turned into RNA. Now, we didn't really talk a lot about RNA last time, but RNA, think of it as a cousin of DNA. It's not exactly DNA. It's got a few changes here and there, but think of it as a cousin. And so DNA gets transcribed into RNA. That process is called transcription. For those of you who maybe have heard about scribes, what do scribes do, right? Scribes write down, they copy, right? They copy, more importantly, they copy in the same language most commonly. So scribes, they take some manuscript and they copy it down into another manuscript. And so same idea here, right? Transcription is the process of taking the DNA and turning it into RNA. And notice it's the same language. It's the language of nucleic acids. Now, when you translate something, you're changing languages. And we'll see what we mean, what I mean when I say that, because when you translate from RNA to protein, you're going into a different language, different alphabet. And some people have mentioned reverse transcription. You can go from RNA back to DNA. It's more rare, but it does happen. And some don't even go on to protein. So again, all kinds of exceptions. But the central dogma is DNA goes to RNA to protein. And yes, we still haven't answered how does this lead to traits. Right? It seems just like we're pushing the problem one step down, right? Okay, we have a DNA sequence that makes a protein. Well, how does a protein determine traits, or how does RNA determine traits, right? And we'll talk about that. And before I move on, I do want to give people a primer on DNA and RNA. So some people might not know what RNA is. People have probably heard of DNA, it's more popular. But RNA is also super important. And so RNA, notice it's just DNA, but without the deoxy, right? So deoxy means no oxygen. Notice that there is no oxygen here. That's a hydrogen, that's an H, right? There's an oxygen here, really small difference. So that's sugar. Remember, a nucleotide has 3 parts: phosphate, sugar, and a base, just to refresh your guys' memories. The sugar is different in DNA and RNA. The sugar in RNA is ribose. The sugar in DNA is deoxyribose. And actually, that oxygen is pretty important because the oxygen makes RNA less stable. It can actually swing around and attack itself to break itself apart. So RNA is a less stable molecule than DNA. Another small difference, RNA typically uses uracil, U, instead of thymine, T. What's the difference? Can anyone spot the difference between thymine and uracil structurally? They're both 1 ring, right? We mentioned last time about how we have 2 rings paired with 1 ring. So they're both 1 ring. Yes. So Ramya said H3C, right? There's a random group here that's different between T and U. A really small difference. And it turns out that difference doesn't actually impact the base pairing. And so that means that uracil, U, pairs with A, just like T. So Remember how T pairs with A, like my initials, Alan Tong. So A pairs with T. Uracil is very similar to T, so U also pairs with A. So A and U are also friends. And of course, the leftover is G and C paired together. All right. Any questions about RNA versus DNA? RNA has U, DNA has T. And again, these are still simplifications because there's all kinds of modifications. But big picture wise, RNA has U, not T. Awesome. So then we get to the language of proteins, right? So we were talking about RNA and how RNA is like the cousin of DNA, but it's the same language, right? Notice how it has the same base pairs. It's got 4 letters. Proteins are a different language in biochemistry because proteins speak in the language of amino acids, not nucleotides. They're a different class of molecules entirely. So amino acids, you might have heard about them, right? If you work out, if you go to the gym, you might, have some protein powder shakes that are full of amino acids. This is what they are. So they're the substituents of proteins. You link a bunch of amino acids together, a chain of amino acids gives you a protein. And that protein, as you can see, can fold in all kinds of really weird helical ways. And this one is actually looks kind of similar to DNA, right? It's a helix, but it's one strand. And what does a protein do? Right. So let me actually move on to here. What do proteins do? Can anyone? Actually, yeah. Let me see if it loads up. I think you should be able to access the link and start answering. So that's the question. What do proteins do? Because we've pushed the question one step ahead, right? We were like, DNA encodes proteins. DNA gives us traits. Well, how? Well, it encodes proteins. Well, how does proteins give us traits? So I'm seeing some answers come in. People are saying catalyze, reactions, work, definitely. They're cellular, right? They're inside the cell. They can also be out of the cell too. They can be extracellular protein. Transport, exactly. Have you guys seen the video of the motor protein? So you have some railroad tracks in the cell, and your motor proteins, literally, they have like 2 legs and they walk along the track inside the cell. I think that's pretty cool. I'm seeing tissue. They can make up tissues for sure. For example, connective tissue is made of protein. Muscle tissue, that's a lot of protein as well, which is why, when you work out, you eat a lot of protein shakes. People talk about those. Regulation. I like that one because not many people will think about regulation, but that is really important. How do we grow? How do we know when our heart beats, or when do our neurons fire? All of that is regulation. You have to control when things happen, right? I mean, if your heartbeat stops, beating regularly, that's called arrhythmia. And so, regulation of things is really important. Control. Everything. Everything. I wouldn't say everything, proteins don't do everything, but they do a lot of things for sure. Inhibition too, so that's part of regulation. Proteins can stop processes, they can inhibit processes. Insulation. I'm seeing more transport. Repair, yeah, exactly. I'm seeing membranes. Cells have a surface, and cells need to control what comes in and out. Inside your intestines, when you absorb food that you ate, that you digested and is flowing through your intestines, you got to get that food, those nutrients, to your cells. You got to absorb them, right? You got to take it through the cell membrane. And a lot of proteins are anchored to allow the travel for those chemicals. And so, as you can see, proteins do a lot of stuff. And proteins are encoded by DNA. So theoretically, you should be able to find all the proteins inside of your genome. Okay, so clearly proteins do a lot. And so you can imagine the complexity of this, right? Let's imagine some trait. Let's imagine, for example, skin color. Why do some people have darker skin, some people have lighter skin? It's because of a thing called melanin, right? I'm sure a lot of people have heard about melanin. Well, how did melanin get there? It's a chemical reaction catalyzed by proteins, catalyzed by, we call them enzymes. And so, a lot of biochemistry goes into a lot of these traits that we have. We know same for hair as well. Why do people have different colored hair? It's because our enzymes can catalyze different reactions to give different pigments, which are chemicals. Right? Life is chemistry. What else? Let's see, build, right? Muscle size. Why are some people bulkier, more swole? Why are some people skinnier? Why are some people's metabolisms faster, slower? It all has to do with these traits. And I'm sure that proteins probably play a role in them. If you have more muscle, you probably have, more myosin, more actin. Those are proteins. And so those are all encoded in your DNA. And environment as well, of course, right? You got to train to get those muscle cells, but awesome. So obviously proteins do a lot. Right? Proteins, they're chains of amino acids, but again, we have to ask ourselves the question, how does a protein do all these things, right? I mean, we talked about it very simply. We said that proteins, they're made out of a string of amino acids. What are these amino acids and what makes them so special? If you look closely, not much actually. I mean, this is kind of surprising, at least to me. People are saying, oh, proteins do so much. They can catalyze reactions. They can make you have muscle. They can give you, certain pigments and certain traits. But proteins are just chains of these. Now, notice there's a lot more letters, right? DNA only had 4 letters. RNA had 4 letters. Proteins, they have 20 letters. And so, as you can see, much more diversity, much more complicated. But again, it's still just chemicals. Bunch of atoms bonded together. And for example, my favorite is this one, alanine, to no one's surprise, of course. But you see, alanine is just some base, some kind of starting point plus a CH3. Notice, for example, cysteine, C. It's the same starting point plus another group, right? So do you see how the highlighted group is different between every amino acid? All amino acids have the same sort of backbone. See that? They're made out of the same backbone. And again, I won't go too much into the biochemistry of amino acids, but just notice, right? Notice that they have the same sort of language. I keep going back to that term. They use the same chemical language, and that backbone is used to link them together. And so, for example, this group might link with this group and form a bond. Right? This group might link with this group, form a bond, and you form chains that way. And you can form these long chains of amino acids. And then, because of the different chemical properties of these different groups, the protein will then fold in a certain way. And that's a really big area of research. It'll fold into a different way. And that structure, that folded 3-dimensional structure, then allows it to do what it's meant to do, right? The motor protein, it's got legs. It has legs that walks on cellular tracks. How did it get those legs? The legs are made from the folding after these amino acids are all linked together. And it's all just chemistry. It's all just, positives and negatives attracting, like charges repelling. It's all just these, really, really microscopic, nanoscopic processes that really come together to give all this complexity that we see in our macroscopic world. Rohan asked, is there a reason why the carboxyl group is sometimes drawn with an is sometimes drawn with an O- instead of an OH? Yes, and so I don't want to get too deep into the chemistry here, but the carboxyl group, which is, for example, the COO group there, Rohan's asking, well, sometimes you see carboxyl groups drawn like this with an H, right, like that. And so sometimes it's there, sometimes it's not. So it's an equilibrium. And it turns out they have something called a pKa. It's just a number, it's a property. And it tells you how often is that H there, how often is it not. And it turns out inside our cells, pH of about 7.4-ish, most of the time it's not there. Carboxyl groups, they have a pKa of about 2, 3-ish. Our cells have a pH of 7. 7 minus 2 is about 5. So about 10 to the, about 1 in every 100,000 has an H. The rest don't have H's. -ish. Good question though. Yeah, it's a chemical equilibrium. Awesome. So this is the language of proteins, right? Link these together and you have yourself a protein. And that protein will then fold into a certain shape. As you can see, at first it's a spaghetti, then it's an ordered spaghetti. And that ordered spaghetti is important for its function. For example, if it catalyzes a reaction, let's say it makes melanin, for example, that chemical, the reactant, will probably fit into a pocket in that folded shape. Right, so that's pretty important. The shape is really, really important. The shape also determines what other things a protein interacts with, right? Why are some proteins in membranes? Why are some inside the cell? Why are some inside the surface of the cell, the membrane? It's again because of how the protein folds. If the protein folds in a way such that it's favorable for it to be in a membrane, it will be transported into a membrane. Well, how did it get transported into the membrane? Other proteins transport it into the membrane. Well, how do those? It's all down to the structure of the protein. Right? So it's really important how these things fold. And this is a static image, but proteins are alive. They move. When enzymes catalyze reactions, they don't just sit there. They can twist and turn, and they can force the chemicals to bump into each other. That's how they make the reaction go faster. So the shape is super important. And so, hopefully you're starting to see how these things can then lead to traits. And the sequence, and I think here's the missing link for those of you who might not have heard about this before. The sequence of the amino acids that determine the fold is encoded by the DNA. Right? Your DNA holds the information for the sequences of these proteins. So, ultimately, it's the DNA that's like the bank, right? And you sort of extract information from that bank to form these proteins. Okay. And again, this is the same slide as last time. 20-letter alphabet, right? A lot more complicated than DNA. DNA, honestly, hot take, DNA is kind of boring. DNA sits in the cell, doesn't do much, right? Just sits there waiting to be transcribed. It's the proteins that do the work. It's the proteins that catalyze reactions, that transport, that form the structures that are very oftentimes found inside and out of cells. Yes, and it all has to do with the shape, right? DNA, double helix. Now, DNA does have complex shapes, don't get me wrong, but I'd say that proteins have a much more diverse and rich sort of shape. And for those of you interested in looking more at protein shapes, this is something I'm really into, this kind of the structural biology. There's something called the PDV, I'll type it in the chat, the Protein Data Bank. And you can go to that website and just look at all the structures that people have solved about the, over the years. Solving the structures of proteins really takes a long time, but it's really a rich source of information. Okay, we answered that question. We know what proteins do. Proteins do a lot of things. And now let's move on to, I'm sorry, this slide is taking me a bit long to move forward. For some reason, these poll everywhere slides just don't want to go forward. There we go. Okay, now here are the two keywords. If you want to take away, two keywords, it'll be these two: transcription and translation. These two processes are really important. Transcription is going from DNA to RNA, as we saw. Translation is going from RNA to protein. Now, look carefully at how this works too, right? DNA is originally a double helix. What happens in order for transcription to occur? The double helix has to be pulled apart. All that glue between the strands have to be separated, like a Post-it note, right? That was evolved. That's no accident, right? Like, why are hydrogen bonds used to glue the strands together? It's because they're weak. You don't want something that's really strong, or else you can't access that information, right? But you also don't want something that's too weak, or else the strands will just fly apart. So you see, it's optimized. It's no wonder that it's hydrogen bonds that hold these together. So first you got to separate the strands, and then RNA gets transcribed. And again, all of this is proteins. Proteins do all this stuff. Okay. And then RNA gets transcribed from DNA by a protein. Now, notice how it's transcribed. It's just like how DNA works, right? DNA base pairs with itself. A is friends with T, right? A is friends with T. C is friends with G, right? RNA has the same rules. A is friends with T, and C is friends with G. It base pairs the same way. One difference though, if you want to see a T, you just change it with a U, because RNA uses U, not T. And so, for example, if our DNA strand was TAC, the corresponding RNA strand would be AUG. It'll be the base pair form. Any questions about that? That's transcription. So transcription is just changing it into RNA. Notice that this RNA strand will be the same sequence as the corresponding DNA strand, the opposite strand, but just all the T's are used. Do you all see that? All the T's just change into U's. And then the RNA flies away. It's like Hermes, right? RNA flies away. He's a messenger. We call it mRNA, messenger RNA. Flies out of the nucleus, goes into the cell, and then it meets a fancy machine called a ribosome, and that's where translation occurs. Translation is going from RNA to protein, and that's where the sequence of amino acids comes into play. So this is a protein. Polypeptide equals protein, basically. It just so happens that the first three, AUG, code for methionine. It's an amino acid. AUC codes for isoleucine, an amino acid, and so on. It's interesting, right? Life reads in threes. I think I have a slide on that actually. Let me move forward. Oh yeah, and so in our cells, human cells, nucleus, DNA is in there. DNA turns to RNA through transcription. RNA flies away into the rest of the cell and gets made into a protein, and that's translation. So bacteria is kind of different, but I won't get into that right now. I do want to get into the code though. We always call it the genetic code. It's like a cipher. Why do we call it a code? It's called a code because it's literally a code. People spent their research lives trying to decrypt this genetic code, and that won them a Nobel Prize, actually. And so literally, people made this sequence of RNA and were like, hey, what amino acid does this make? How about this? How about this? How about this? And they figured out that life reads in threes. We call them codons. They're triplets, right? So 3 bases code for a single amino acid. For example, UUU codes for phenylalanine, Phe. It's an amino acid. So if the machine sees UUU, that's code for, hey, use this amino acid called Phe. If I see AUG, that's code for, hey, add a methionine onto there, add met onto there. Right? It's literally a code inside of our cells. And what's even more striking is this code is pretty much constant throughout all of life. Now, it's different in some, it turns out, but for the most part, we call it the universal genetic code, but it's not really universal. But let's say it was. What does that tell you about this code? The fact that pretty much all of life has it. Here's the observation. We notice that almost all life we see uses the same genetic code. What can you infer from that? And Rohan said that it evolved early. And I would definitely agree, right? It evolved early and hasn't really changed. It's through evolution. It's really, really early because if it was recent, then that means that some would have it and others won't, right? If it evolved early and then all the ancestors are life as we see it today, then most likely it'll have some sort of variant or some similar code. And it turns out that it's really important that you can't really compromise this code, which is why it's been relatively conserved. Awesome. Okay. Any questions about the genetic code before I move on? Let's move on. A fun thing for you. So, we were talking about genetic codes. I've written my own code. This is my code to you all. Why don't you all give it a try and try to decode what I'm trying to say here? So I'm giving you a sequence of DNA, and your job is to be the ribosome, be the machine, and take the resulting RNA transcript and translate it into a code that you can understand. And so I'll just give you, the first one to sort of lead you all on. I see the first three, because we said life reason threes, right? I see TCG, AGC. I haven't really talked about this, but DNA is directional, and so that means that only one strand is really used. Turns out that it's the TCG, it's the 3 to 5 that gets transcribed. And so transcription, we know our rules, right? T is friends with A, C is friends with G, G is friends with C. So I see, oh, AGC, that's the RNA. I've transcribed it into RNA. What letter is that? Oh, AGC on the ribosome. Now I'm translating this code, AGC, 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 that's here, AGC, and that code's for serine. It's an amino acid. And the one letter code we use for serine is S. And so I'll just write S here. These first three stand for S. And I want you all to do the rest. So tell me what I'm trying to say here. And once you have it, text it privately in the chat. That would, you don't give it away to everyone. So text it to me. Is the process clear? Any questions? Please ask. We're transcribing, we're translating, and we're decoding the DNA. Well, my DNA. This is my DNA. It's not real. Good. And once you have your sequence of amino acids, use the one letter code. Text it to me privately in the chat. This does take a while, right? So take your time. And I got a question. A does indeed pair with U. Yes. Yep. So I can do the next one. Or nice. So some people are showing their answers in. Awesome, awesome. But yes, so A pairs with U. So for example, this one, that would be a U. Nice. I'm seeing answers rolling in. Awesome, awesome, awesome. Yeah. So, now you're seeing the code aspect of it, right? It's literally taking, one language and turning it into another. But how long does that take you guys? Like a minute and a half? This would probably take a ribosome probably less than a second. Right? So just crazy fast. Or, a couple seconds maximum. So ribosomes are crazy, crazy fast chemical machines. And you think about it, all of this is just a chemical reaction. You're linking together amino acids, you're reading the transcript, and it just happens extraordinarily fast, faster than we can do it. Awesome. And so I will move on. For those of you who are still working, I'm sorry, I think I will have to move on. But I hope you had fun with that activity. The answer was stay safe. Right? So notice, we have S, serine. T was, I believe it's threonine. Where is it? Threonine. Yep. A is alanine. Woohoo. Y is, where is it? Tyrosine. Right? And so it's a sequence of amino acids. And these are all linked together. So, I did neglect a few things. So typically inside a cell, you have what's called a start codon. So pretty much most things would start with this red one here, AUG. And you got to know when to stop too, right? So there are stop codons too. So if a cell sees UAA, it knows to stop right there, cut it off. And so then it truncates the peptide in the fold, you have your protein, it spits it out. Okay, any questions about transcription and translation? Because these are really important concepts. The idea that cell takes the information in DNA and does it and uses it to actually do something. It makes proteins that do something. So I think I got a question. Is the start codon translated? Yes, good question. Methionine is typically the start of a lot of proteins, and you would typically then have some sort of modification. So some proteins might cut it off, some might modify it in some way. Bacteria actually use a different form of methionine. They use formal methionine, not methionine, but we use methionine. Eukaryotes use methionine. So you can see there is some variation. So yes, and in short, yes, it is translated. Awesome. Cool. Now let's get to the real sort of heart of what we're after. We're after traits. We're after how does it work. I mentioned pigments. I mentioned melanin, right? We have melanin in our skin, in our hair. That gives it pigment. We can see color. It absorbs certain wavelengths of light, so we can see, for example, ginger hairs or blondes or black hair like me, right? Things like that. Flowers too, right? So there might be some DNA in the plant, and the plant cell nucleus holds the DNA. The DNA is transcribed into RNA, and the RNA is translated into protein, and that protein, after it folds, might make a purple pigment. And that might be why the flowers you see outside are purple or red or maybe white if it lacks pigment, or maybe there's some white pigment, who knows, right? So you can see how proteins can give rise to traits. If any of you have heard of, Mendel's experiments with the pea plants, where he looked at the colors, right? This is why. It's because the pigments were made by proteins encoded by the plant DNA, and so when he crossed the plants, they could have variation in their pigments. So, trait 1, right? Colors. Other traits. Does anyone know what this is? Anyone recognize this molecule on the left? I'd say it's a pretty famous molecule. It's kind of interesting. It's got a square right there. And you'll notice I'm very much a chemistry dude. Colin might be more on the bioengineering side. I'm more on the chemical side. Anyone have a guess what this is? Or what it does, or function-wise, like what it is? Any guess is fine. This is a steroid. Good guess. I mean, you recognize the rings, right? So steroids have rings, but it's not a steroid. That's a really good guess, though. I appreciate it. This is penicillin. So penicillin is an antibiotic. It's something that kills bacteria, which is awesome, right? I mean, back in the old days, when you got a cut, it was bad, right? If you got infected with a cut, it was like life or death. But now we're like, antibiotics, we can kill the bacteria. So now cuts aren't dangerous anymore, or as dangerous anymore. Penicillin was one of the first antibiotics discovered. And so when people, got injured, they want to prevent infection, they would use penicillin. They would use this molecule. Bacteria are smart. Bacteria, they evolved the DNA sequence, which is transcribed into RNA and translated into a protein that's called beta-lactamase. What does ACE mean? Anyone know? It's an enzyme, exactly. ASE, that ending, if you see that probably means enzyme. It makes reactions go faster. Beta-lactamase is an enzyme. It's a protein, exactly. It's an enzyme that does this reaction. What did it do? It got rid of the square. The square! The square is the most important part of the molecule. The square is where the antibiotic actually attacks the bacteria. The bacteria, penicillin, it's what's called a cell wall synthesis inhibitor. And it does that because the square is so reactive. The reactive square allows this antibiotic to get into the cell wall and it activates some enzymes. If the enzyme breaks that square, suddenly the antibiotic doesn't work anymore, right? What does that mean? It means the bacteria is resistant. Y'all, this is antibiotic resistance right here, right? This is an example of how DNA can code for a trait that might not be visible, right? antibiotic resistant is a trait that's coded, for example, here by DNA that encodes for this enzyme, which is a big problem, as you can imagine. I'm really fortunate to be doing some work on antibiotics this summer, so this is why my mind is very antibiotics. But anyways, another thing is diseases. So when you talk about traits like, oh, eye color or hair color or 5 fingers, those are interesting for sure. I mean, you can study hair color, but I think of more interest to a lot of you might be the medical side, the clinical side. When people have, for example, here is cystic fibrosis. Their airways are clogged. It's really hard to breathe. They have a really hard time getting oxygen because of this disease. And guess what? It's a genetic disease. That means that it's in your DNA, which is scary to think about, right? I mean, you have a sequence in your DNA that gets transcribed into RNA, that gets, I'm repeating myself, transcribed into RNA, translated into protein, and that protein, in this case, folds into a transporter. It's a chloride transporter that sits around at the surface of your cells. And it turns out, if you have a malfunctioning chloride transporter in your airways, mucus builds up. And you can imagine mucus inside your airways, if that gets really thick, it gets cloggy, it's really hard to breathe, right? So it's a really horrible disease. And so, the thing is, it's a genetic disease. What if you can modify the DNA to change the protein to make it back normal, right? Gene therapy, right? Again, DNA is a chemical, and chemicals are atoms bonded together, and we can modify atoms through chemistry. So what if we can modify the DNA? Okay, that's the whole idea behind gene therapy. Again, would you do it? I would say probably yes, right? I mean, this is a really clear case of a debilitating disease that is fixable theoretically. But again, of course, as a lot of you have probably heard, there's a whole field of bioethics, right? Should we or should we not edit the DNA? And the argument is, well, some people say that, oh, well, if we use gene therapy, if we modify DNA at all, it'll just blow up and people will start modifying themselves to be superheroes. You can give yourself super intelligence. You can give yourself maybe you can slow aging. You can give yourself bigger muscles, leaner muscles, whatever you want, really, right? Assuming you know the genetic pathway. Of course, it might not be simple, but it's theoretically possible. I would argue that the use of gene therapy in things like cystic fibrosis, where these individuals literally have this really horrible condition that they have to live with, I'd say that's ethical, right? It'll be unethical not to use it, in my opinion. Again, this is more murky area because this is very new. But again, that begs the question, where is the line? I'll leave you all to ponder that, because we need people like you who are interested in this to think about these problems and really deal with these issues ethically in the future. Okay, let's move on. We talked about one idea of engineering. We talked about the engineering of possibly the cystic fibrosis protein to make it normal again. What else would you want to engineer? Can you think of something else that you would be interested in? And again, this might be prompting you to think about the final project. What are you interested in? What might you engineer in cells? It can be any level, DNA, RNA, protein, or whatever you want, right? What do you want to engineer in cells, in living systems? So much cancer cells. That's a good one, right? If you could engineer cancer cells to not be cancer cells, that'd be amazing. Viral protection. Yes, exactly. I was, last summer, actually, I was working with something called AAV. Now, I wasn't working directly because it was COVID time, but I was modeling AAV. And it turns out that you can use this virus, this AAV virus, to deliver DNA into cells and have the cell synthesize antibodies. Imagine that, right? A gene therapy for immunoprophylaxis, to give yourself antibodies without, it's like a vaccine almost, right? Bacterial resistance, right? If we could engineer ourselves to have bacterial resistance, then maybe infections are a thing of the past, right? Or we can engineer bacteria to not have resistance, in order to, make them more susceptible to drugs, or a lot of them we actually want to make them resistant in order to select for certain ones that you might see in certain classes in the future. More cancer, right? Exactly. If we can detect a cancer early, why not modify it, right? Agriculture, yes. The idea of GMOs and crops, that's a really big issue as well, right? There's a crop called golden rice, which is rice that's yellow because it has vitamin A. Would you eat it? I mean, I'd say that those of you, not those of you, those individuals who are living in perhaps places that might not be as nutrient rich, I would probably, want to have more vitamin A in those regions. So that's important as well. But yes, pest resistance as well. Personalized medicine, yes. In the future, it's many people's dreams that we don't just use our sort of crude, hey, this worked for some people, will it work for you? Try it out. What if we could personalize medicine, sequence your genome, and I look for certain tags and I say, oh, this will work for you or this will not, right? There's certain subsets of genotypes that might work for some drugs and not others. So that's a really good point. Plastic degrading bacteria, I love this one. Moving on to environmental sciences, right? We have this issue of landfills and plastics building up. What if we can engineer a plastic eating bacteria? And those I think exist actually, but what if you can make it better? Or, have different types of plastic eating bacteria, all really good things. Reverse antibiotic resistance, exactly. More cancer cells, cell-to-cell communication. All these are really good ideas. And someone said in the chat, it's Alzheimer's disease. Exactly, so more treatments, more clinical, right? If you could have, if Alzheimer's disease is genetic in a certain way, maybe you can modify it. Copying a CRISPR-like system into eukaryotic cell. Oh, interesting. So CRISPR, for those of you who don't know, is a gene editing technology inside bacteria. It's a bacteria immune system. And what if we could use that and put it into eukaryotic cells? I have good news for you. That's been done. People have CRISPRed eukaryotic cells before. Now, of course, it's the bacterial version of CRISPR, but you can use it in eukaryotic cells. So that's definitely a good engineering point. So you can see, it's so applicable. Literally, anything you can think of, you can probably engineer, right? This field is so powerful and it's so rich. And you can just see, the range of responses that we got and the range of interests in this group already speaks to the diversity in this field. And I have no doubt that a lot of you will go on and do great things and do these things in the future. Blood cells to transform its interactions with other cells. Exactly. So a lot of blood groups, A, B, or O. I'm A. I'm blood type A. Blood transfusions. You have to be really careful about which blood types you can transfuse. What if you could engineer an enzyme to cleave off those groups to make all blood types transfusible? That'd be amazing, right? I mean, in that case, the what's it called? The blood banks might not be as in need of supply. So that's a really big thing as well. Delaying senescence. Yes. So senescence, for those of you who don't know, is like cell aging, essentially. It's like when a cell just stops dividing and growing. It's just sort of dormant. If you can delay senescence, what if you could slow aging? Right? Is there an aging gene? There are these things called telomeres at the ends of chromosomes that shorten as you age that we observe. What if you could replenish those? Right? So all these really sort of sci-fi things are suddenly within reach with bioengineering. So you can see what Colin means when he said this is the future, because it is, right? This is the future of bio and bioengineering. Now, of course, you can think about issues with all of these too, right? If you delay senescence, if you activate cell division, might that increase cancer? My guess is probably. So you got to be really careful with these things. If you transform blood cells to make them more transfusible, or changes interactions, who's to say you might not increase some other immune response? Also a good question, right? So again, bioethics plays into each and every one of these. Even this one, right? Plastic degrading bacteria. Awesome, right? We got plastic landfills. Why don't we just throw the bacteria at them and have the bacteria eat these plastics? Well, what if the bacteria also affect ecosystems in certain ways, blah, 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 blah. You can see there's a lot of thinking that goes into each of these projects. So I wish you all the best of luck in the future. All right, let's move. Oh, shoot, what time is it? Let me check. Oh my goodness, I have 8 minutes. Okay. I always do this. I always ramble for too long. Anyways, for the last 8 minutes, I want to destroy everything I just said because I have told you that the central dogma is DNA to RNA to protein. Guess what? It's not so central, is it? 1.5% of your genome is used to code for protein. Not so central, is it? A big part of it, and people call this, the dark matter of the genome. I think Colin brought it up last time. A lot of it's unknown. A lot of it is just like random repeats. A lot of it comes from viruses. So viruses sometimes infect our cells and they just stay there. Their DNA is just in our genome. A lot of it is transposons, random sequences that just kind of jump around, they hop around. So again, a lot of really, this is the vaguest label ever, miscellaneous unique sequences. What does that even mean, right? A lot of people don't know. And so again, I leave it up to you guys to figure it out. So a really big, and again, what we talked about just now, a lot of it can be done with this 1.5%. Imagine if we understood the other, 98.5%. Imagine the power we have for biology then. So big ideas, big ideas. Oh, I see a thing in the chat. What are signs and lines? Oh, good question. In the previous slide, the signs and lines, those are viral sequences, I believe. I might be wrong on that, but I believe they're viral sequences that have integrated into our genome. And again, I'm not sure what they do. They might have functions, to the Google. So, yep. And feel free to Google and tell us next time. Okay. So yes, we've talked about sequences. We talked about DNA sequences, how they code for RNA sequences, and how that codes for protein sequences. And we also talked about how a lot of DNA sequences we just don't know. A lot of it's regulatory, controls things. But again, a lot of it is really not well understood. But obviously the sequence is important. And so I want to spend the last sort of 5 minutes just talking about how we actually know the sequence, because I want to go back to experiment. We talked about big picture things today, right? The exciting things about biology. You got to do experiments to figure things out. So how do people sequence DNA? This is one of the first ideas that people had. This is called sequencing by synthesis, or Sanger sequencing. This dude called Sanger came up with it. The idea is, I've got this DNA sequence. I want to know what it is. Imagine I don't know what this is now, right? But sort of use your magic and just you know it now, right? So how do we figure out what sequence this is? Here was this idea. What if we could sequence it by synthesis? We can make the DNA, because as we know, DNA, it can base pair. So for example, this C base pairs with this G. This T base pairs with this A, G pairs with C, etc., right? We can make the other strand of the DNA. And we can introduce special bases, special nucleotides, actually, that halts synthesis randomly. So here's the idea, right? Let's say you add in your special G. So here are the special Gs. So the G just halts the synthesis. It doesn't have the O, actually, the oxygen. But that's details. The idea is this: when you make the DNA sequence, it'll make, and so at some point it'll just stop because it encounters a special G. So it might stop here when it makes the first G. It might stop here when it makes that G there. It might stop there when it makes that G. You repeat with each of the 4 letters, G, A, C, and T. And you can see you get a whole, a whole sample of sequences. And more importantly, you can separate these out. There's something called a gel. And you can see that this gel is able to sort the sequences by how big they are. So of course, if you run into a G at the beginning, a special G in the beginning, and you stop, that's going to be a short sequence. It's going to be pretty short. And so you can see the G one goes all the way down to the end. It's small, right? It's short. Whereas a really long one, the T, right here, all the way, this is the longest sequence they made. It's pretty big, right? So it's up here. So that's that one right there. Anyone know why the smaller ones travel farther down? What's the logic behind that? This is like a gel thing. You have a group of DNA, you put it inside. Why do the smaller ones travel to the bottom? So someone guessed density. Ramya said they're able to travel faster. Yes, faster. Why? It's not density per se. So someone said less mass. That's true. That's true. Someone guessed electric charge. That's also true. So DNA is negative. So there's positive here. There's negative here. So obviously the DNA goes from top to bottom. So it runs down. The smaller ones run faster. Why is that? Yes, exactly. So someone said it. It can fit through smaller pores. So, you know, my family, no, not me, because I'm away from my family. My family went to like a park the other day, and then in the park they had like this playground thing for little kids. My brother, he's 8. He can crawl through the hole really easily, the tunnel really easily. And my dad followed through. He had some trouble going through the kid hole. And so same logic here, right? The bigger ones have a harder time traveling through the pores in the gel, which is why the smaller ones go down further. So you can see really easily now we can just read off the sequence, right? We have at the top, we have T, then C, then G, then A, and all the way down, etc. Right? So this is sequencing by synthesis. We made the DNA, we separate the DNA, and now we can just simply read off what this is. T, C, G, blah, 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 blah. And then to get our original, we just base pair. Right? So that's our original. So this is like the OG way of sequencing DNA. As you can see, a lot of work, a long process, right? Gotta make it, you gotta run the gel, then do some analysis, long process. Time do I have? 1 minute left. Okay, last closing thought. Now it's a lot easier. Now it's a lot easier. There are companies to do this for you. There's, things like, there's companies like Illumina that can sequence DNA for you. They can use, fluorescent dyes, colors that correspond to each base pair. You can even have what this is called an Oxford Nanocore. You feed a sequence of DNA through it, and depending on the current running through that pore, you can measure out the sequence. It's pretty cool. So now there's all kinds of fancy ways to sequence things. So that was the old way. It's pretty logical, right? But now there's a whole, revolution of DNA sequencing, which is really important. And so in your projects in the future, if you want to engineer things, I bet you'll use DNA. You'll probably use one of these techniques. You'll probably sequence DNA to see exactly what letters you have. Okay, I will end it there. And if you need to go, you're free to go. Thank you all for listening. Hope you had fun. I'll stick around for any questions now. But yeah, happy 4th of July, everyone.